So it is my pleasure to introduce Dr. Jim Estes from UC Santa Cruz. Jim got his PhD from University of Arizona, and then he immediately started working for the Fish and Wildlife Service. And he came to Santa Cruz when it was the Fish and Wildlife Service in the late 70s, and he's been there ever since. And he followed the name through after became part of the USGS. And I think I was one of Jim's students during this time, and we used to be the the National Biological Survey, or National Biological Service, and then we became the Biological Resource Division of the USGS, where he was there for up until about 2007, when he retired from the service and then took a position as a faculty member at UC Santa Cruz. Jim's work, we'll talk about some of that today, has kind of become a household name in a lot of ecology groups, particularly with looking at trophic cascades and things like that. And so much so that one of the things that's happened this year is Jim has been inducted into the National Academy, which is a huge honor, as you can imagine, and all the festivities and all that happen, I guess, in the coming months. So one of the things that Jim, I'm kind of hoping that Jim will talk about, he'll have his own talk today, is kind of, what's a historical perspective? What's it like to work in a system for decades at a time, and the insights that come over thinking about things for long periods of time. So many of us go from one thing to the next, to the next, to the next, and how often do we actually get time to sit back and work in a system for just, for huge portions of our career, and the kinds of insights that come from that. It's really been exciting from my perspective to watch that, and I'm very grateful for Jim for exposing me to the Aleutian Island, that ecosystem, where I was working in Mexico for a long period of time. I got introduced to the Aleutian Islands, and since then I've been going back up, and I'm heading back up with my colleague Brenda Konar and I for another couple of cruises. So that's been a fantastic way to get introduced to that system. So without further ado, I'm going to let Jim talk about his ideas on what's happening for the past several decades of his career.
Well, thank you very much, Matt. Kind introduction, and thanks you, all of you, for coming. Actually, one of the first seminars I ever gave in my life was here. Mike Foster invited me up when we were both young, and I just moved to California, and I came down to Moss Landing for a seminar. I bet Greg, you probably don't remember the seminar, but you were around then in those days. And so I've been back here a few times since, about every 5, 6, 7, 8 years it seems, and it's always great. I always have a good time, and wonderful place. You guys have just done a phenomenal job in educating people, and all of the great people who have come through this program, the graduate students. It's something to be very proud of. Okay, so what am I going to do today? Well, it should be fairly obvious looking at me that I'm closer to the end of my career than I am at the beginning. And as a consequence, I'm going to give something of a retrospection, all right? Something of a view of the things that I have done over the course of my life. This is pieces of a book that I have just finished. It's going to be coming out hopefully in another 6 to 8 months. And so some of the modules of that book are really going to be the substance of my seminar today, all right? So I need to start out by providing something of a conceptual framework for the way I look at nature, all right? And this is actually a figure that was in a paper that was published in PNAS back in 2005 by Jordi Viscompte, a Spanish theoretical ecologist, on understanding food webs. And it's a characterization of a food web. So all those little circles around the outside are just figuratively species, and the links between them are, the arrows between them are the linkages between species. That is the consumer-prey interactions, the weight of those. This is actually a Caribbean food web that was put together from some empirical data and a bunch of modeling and a whole bunch of other things. It's interesting because it's one of the first efforts to really put together a food web that characterizes interaction strength both in terms of directionality and weight. So those different arrows are indicative of the strength of interactions in this food web. So when we look at nature, we see species. They're easy to see. And we see how abundant they are and where they occur. That's a relatively easy thing to do. And we can also characterize food webs by watching these species and determining who eats whom. And if you do that long enough, you can put together the topology of a food web. My argument is that means almost nothing. It gives you a topology. It gives you a roadmap but it doesn't tell you anything about the dynamics of those arrows. And what it is that, at least by my view, that we would like to do as ecologists is understand that dynamic infrastructure. Dan Jansen had a great quote in a paper he published 30, 40 years ago about extinctions, and he made the comment that species interactions are invisible. They're invisible unless you do something to see them. So you have to somehow perturb these species in order to see that infrastructure. And that's what experimental ecologists do. And I'm not really an experimental ecologist, but I consider myself sort of a perturbational ecologist, and that's what I've tried to do. So that just gives you a sense. Now let me go all the way back. That's the way I see the world now, in a way, just in a nutshell. But let me go back to the beginning. This is the way I saw it in 1970 when I first started working at Amchitka Island. There were sea otters, and they lived in a coastal ecosystem, and my view of nature was that somehow that coastal ecosystem was nourishing these animals, and that was what we needed to learn about. And then just very fortuitously, I met this guy. That's Bob Payne. That picture was taken in late 60s when he was working with Tom Zarrett down in Panama on peacock bass. And I met Bob, and he said, I was telling him he was up there on a student committee, and we sat down and had a beer one night, and he was saying, what are you going to do for your research? And I said, well, I'm going to do what I just showed you. And he said, God, that sounds dreary. Why don't you do something interesting? Why don't you think about the system from a broader perspective? And in particular, have you ever thought about what these predators, the sea otters in this case, who's out at Amchitka Island, what they're doing to the system? And, you know, my mind just kind of lit up because I thought, well, I don't know that they're very important, but it would really be easy to look at. And it would be easy to look at from this perturbational perspective because of the history of the system and the situation that existed out there. So we had at one time otters spread all across the North Pacific. The Pacific Maritime Fur Trade came through and virtually wiped them out. And then a few of the surviving colonies recovered. And so when I came along on the scene in the early 1970s, there were places that had recovered completely, as far as we could tell. The otters had recovered in these places, and a lot of other places where they hadn't recovered. And so it was a pretty simple matter of just going to the places where they'd recovered and comparing those with the places they hadn't recovered. And that seemed like a cool thing to do, and that's what I did. So here's kind of a schematic of that showing the geography, just to ground you on where we're talking about. It's where we were working out in the Aleutian Islands. These red dots are places where remnant colonies survived the fur trade. The blue dots are places where animals were reintroduced in an effort to get them reestablished because they were completely exterminated over this entire area. There's our California population. There were a couple of others. There was a surviving remnant colony at Amchitka Island, and then over here in the near islands, they had been completely extirpated. And so in the 1970s, when I came on the scene, I went here and looked at Amchitka Island, and then I went over here and looked at Shemi Island, and this is what I saw. At Amchitka Island, I saw lots of kelp, very few urchins, and when I went to Shemi Island, there were urchins everywhere and really very little kelp. And it was pretty easy to figure out what was going on because I knew that otters ate urchins and I knew that urchins ate kelp, and so the obvious explanation for this, right or wrong, I wasn't absolutely sure, but the obvious explanation was that we had this sort of top-down forcing function, this thing that has subsequently come to be known as a trophic cascade. And so when otters were abundant in the system, they limited urchins. That allowed kelp to become abundant. When otters were removed, the urchins became abundant. They ate all the kelp. So it was just about as simple as that, at least in our mind. So I think that's true. I think it's true in a lot of places. We've seen it over and over again. But there were a lot of other questions that began to emerge from this simple model view of the way the system worked. How general was it? Okay, did we just go out and find a place where we saw what we wanted to see and compare it with another place where we saw what we wanted to see? I worried about that in the beginning. So characterizing the generality of the interaction over large scales of space and time became a huge challenge and something I spent 30 years working on. These systems are very different depending upon whether otters are in them or not. When they're gone, they tend to take a configuration that's been referred to as an urchin barrens, a pretty different looking place than when otters are there. You have a kelp forest. How do they transition from one to the other? And what are the dynamics of those transitional processes? I was interested in that. Do they just gradually transition from an urchin barren to a kelp forest as the otters recover? Or do they transition in some other way? What are some of the broader effects of this trophic cascade on the ecosystem? And that was an obvious question because plants matter, autotrophs matter. We've got a system in which the abundance of autotrophs varies remarkably depending upon whether the predators are there or not. So it stands to reason that you might expect there to be some knock-on effects. If these interactions are strong, and they appear to be very strong to me, and if they played out over sufficiently large scales of space and time, one would expect some selective consequences of this. That is to say, you would expect to see evolutionary fallout. So that's another dimension to what we have done, and I'm going to tell you a little bit about it. And then lastly, and completely unexpectedly, is that we discovered fortuitously toward the end of the period of time that I've worked in the Aleutians, that is over the last 20 years, that in fact these systems do not function as spatially isolated entities. That is, a kelp forest is really not a kelp forest. It's linked to the land and it's linked to the sea in super important ways, and of course people have always known that, but what we've discovered is that they're linked together by the movements of large mobile animals. These are really important connections, so I'm going to end by telling you a little bit about what we think is going on there. All right? So firstly, generality. What is the issue? Well, the issue is, I've shown you some pictures of what a kelp forest looks like when otters are there, and I've shown you a picture of what it looks like when they're gone, but how widely occurring is this over the range of the species? So the way that we have approached that problem is that we've picked areas. This is the Aleutian Archipelago, this is Southeast Alaska, this is British Columbia. We've gone into these places, and with an a priori knowledge of whether otters were abundant or absent, but no a priori knowledge of what the reef systems look like, we've sampled these reefs as randomly as we could so that we could infer from that sample that the patterns that we saw were representative of these larger scales of the ecosystem. So here's how we did it. This is just one of the islands. It's probably not even one we actually sampled, but I just got a picture to show you what we did. So we superimposed a grid, just a square grid over the island, over an image of the island. We do this all electronically now, but in the old days, we just put a grid over an island, a map of the island. We took all of the intersection points as potential sample points, and then took a random sample of those intersections and actually sampled those sites, and then took random samples as best we could of the seafloor at those sites. So that gave us what I think is arguably a representative sample without any reason for any sort of a priori bias in what the data would look like. And we did that a lot over a lot of places and a lot of times. And here are the data. So these are data from the Aleutian Islands, and the black symbols, well, first of all, the vertical axis is the abundance of kelp. The horizontal axis is the abundance of sea urchins. The black symbols are representative of systems where otters were at or around carrying capacity. The open symbols are representative or come from systems where they are absent. There are none there at the time that this was done. And as you can see, the data look pretty different. They aren't point estimates. It's not one way at a place when otters are abundant and some other particular way at a place where they're absent. There's a lot of variation. But in general, what we see is very little variation in urchin biomass when otters are abundant. They're uniformly low. Quite a bit of variation in kelp biomass when otters are abundant. They're high but variable. And sort of the flip side of that is that when they're absent, we see virtually no variation in kelp biomass, very low generally, and quite a bit of variation in urchin biomass. So that is a characterization that actually, I think, properly generalizes the degree to which this trophic cascade operates in that part of the world. Now, these are just data from the Aleutian Islands. So we did the same thing in Southeast Alaska. A little bit less data there, as you can see. Pattern's actually stronger in Southeast Alaska than it was in the Aleutian Islands. And then here's some data from British Columbia. And I need to explain these a little bit. These are data that were gathered by Jane Watson, one of my former students, in a 20-year study that she did along the outer coast of Vancouver Island. She worked in 3 areas. Barkley Sound, which is down near the south end of Vancouver Island. There are no otters there, and there never have been during the time that she worked there. Cheklesset Bay, which is up near the north end of Vancouver Island, and there have been otters there the whole time of her study. And then Cayucet Sound, which was invaded by otters during the 20 years of her study. So, if you look at the data from Barkley Sound, otters absent. Sorry, the axes have been transformed here. I cribbed these out of the paper. But basically it's the same thing. We have kelp on this axis now and urchins on this axis. And you see basically the same thing. You see the same patterns of abundance of plants and herbivores depending upon whether otters were there or not. And then at Cayucet Sound, over the 3 periods that she sampled it, 1988 when otters were absent, 1994 as it was being invaded, the gray dots, 2007 after it had long been invaded, you see the transition from one side to the other. So we've got a lot of information to show that over relatively large areas, this trophic cascade that dictates the abundance of urchins and plants in the system is fairly generalizable, all right? So how do these things switch from one to the other? It's a kelpy place, it becomes an urchiny place if the otters are lost. Or it's an urchiny place and it becomes a kelpy place if the otters recover. And how does that happen? First thought, it probably happened in a fairly gradual way. That is just a linear transition. As otters became more and more abundant, they ate more and more urchins, and kelp became more and more abundant. But these are all the data that we've ever gathered from the Aleutian Archipelago over all the years that I've been working out there. So each one of these points represents a mean from an island and a time, all right? So there are a lot of islands. There are actually 19 islands, quite a few different sites. And the densities of sea otters in this data set range everywhere from 0 up to superabundant, that is carrying capacity. So we have everything from otters absent to otters present and everything in between. And the pattern of the distribution, the covariation of the urchins and the kelp is exactly the same as it is between systems where they're completely absent and very abundant. So that made me realize that these systems must transition very abruptly. You don't have intermediates. There are no intermediates. We don't see any intermediates in the data here at all. So that made me realize, this is actually a figure that I copied out of Martin Scheffer's book on critical transitions in nature. It doesn't happen like this. As otters become more abundant, the state of the ecosystem doesn't go from being an urchin to a kelp-dominated state gradually. It happens pretty quickly in some way, okay? So in order to actually do some more analytically, some more sort of quantitatively rigorous work on this, what we needed to do was to have a way of characterizing whether the system was in a kelp-dominated state or an urchin-dominated state. So we just did it this way. Plotted all our data. These are the data basically averaged on islands in time now. We did a cluster analysis, and sure enough, the clusters fell out as we would expect. One cluster, which is this urchin-dominated state. The other cluster is the kelp-dominated state. We simply used a discriminant function to define the two, to give us a quantitative metric. So in any system that we measured, if it fell on this side of the discriminant function, we called it a kelp-dominated state. If it fell on this side, we called it an urchin-dominated state. Then we could go back to all of our data and look at ecosystem state as a function of otter abundance. And so we've done that. And we've actually done it in 2 ways. We've done it for systems that were recovering, that is going from places where otters were absent up until they became very abundant. And we've done it in systems that have collapsed. And I'll talk about the collapse later. But for now, just realize that what's going on in this system is that the otter numbers are different. This is actually a logistic fit to these data. I couldn't fit a logistic to the blue data because I never saw the system actually shift to a kelp-dominated state. This is from Attu Island. And before it could get there, the whole Aleutian Islands fell apart. And so I never was able to actually follow it. So these are just a connection of the dots. But what you can see here is that there are radically different trajectories in these phase states. That is, between this 0 state where urchins are abundant and kelp are rare, and the one state where kelp is abundant and urchins are rare, that the trajectories of change are very different depending upon whether you're going up or down. In other words, this is a system that is characterized by classic pattern of hysteresis. All right? So why is that? We've spent a lot of time trying to figure out why that is. And basically, the most important player in this lesson was Brenda Konar and her work. And Brenda did something that was really brilliant. And she said, we're trying to understand this. You don't take us lifetimes to watch these things change in time. Let's look at them in space. Because when you go out there and dive on these kelps, on these reefs, you see patches of kelp here and there, even in the urchin-dominated places. And so what Brenda did was use this sort of space-for-time approach. She looked at places where there were patches of kelp and patches of urchins, and she looked at the boundaries between the two and studied the transitional dynamics in space instead of time. So this just shows some data from a bunch of different sites within a barren ground to a kelp bed. And as you can see, just like I have argued that these systems transition very abruptly, in space they transition very abruptly as well. All right? There's just urchins and brown algae and red algae, and you can see that it happens very quickly. So Brenda actually did most of this, did it all, did some experiments. And the experiments is just sort of a funny little cartoon that I drew. I'm actually very proud of this because my artistic skills are very limited. So this is a characterization of this transition, kelp, urchin, all right? So in the kelp, at the kelp edge, at the kelp border, we did two things. Added urchins to see what happened, added urchins to see what happened to the kelp, removed kelp to see what happened to the urchins. And over in the urchin side, we did the converse. We added kelp to see what would happen to the urchins, and we took urchins away to see what would happen to the kelp. All right? And what we found was really very interesting because it's not a symmetrical relationship at all. When you add kelp here, the urchins go away. They actually disappear. So when it's in an urchin-barren state and you add kelp, the kelp actually has an inhibitory effect on the urchins, and it does that just through the physical interaction of the sort of the labile, very flexible morphology of the kelp plants and the water movement. So the water's just taken these kelp plants and beaten the urchins away, and that's what happens. All right. Similarly, when you remove the kelp here, the urchins invade. All right. So this just shows the nature of the species interactions between kelp and urchins when you're in a kelp state, the dark arrows, and when you're in an urchin state, the dashed arrows. When you're in a kelp state, it's a, when you're in an urchin state, okay, it's a classic agonistic consumer-prey interaction. That is, the prey has a positive effect on the consumer has a negative effect on the prey. Classic plus-minus agonistic interaction. However, when it's in the kelp state, it's very different. The kelp has a negative effect on the urchins. The urchins essentially have no effect on the kelp. So it's a mensualistic interaction. For all of you that took general ecology, very different. Same species, same place, slight difference, whether kelp is there or not, or whether urchins are there or not. The nature of the interactions are fundamentally different. And this, I think, explains a lot of why we see this hysteresis. Depending upon the state that it's in, it tends to stay there, and once it shifts into another state, the nature of the species interactions change, and that tends to hold it in that state. So that's why I think it happens the way it does. All right, now I want to move on and talk about these so-called broader knock-on effects of the otter-urchin kelp trophic cascade. So we've got this interaction where otters influence urchins, urchins influence kelp, kelp provides habitat, kelp is an important source of primary production, kelp therefore ought to do a lot of different things to other species and ecological processes, and we've looked at a few of those, and I'm just going to tell you about some of them. We've looked at the effects on eagles, the effects on gulls, the effects on one group of fishes, the hexagrammids, the greenlings, And we've also looked at the effect on carbon dioxide uptake from the atmosphere. So I'm just going to run through a few of those things with you. And what do we do? How do we do this? Well, we did it just like we did the earlier stuff. We just went to places where otters were abundant, went to places where they were absent, and looked at different species and what they were eating or whatever we were interested in, and contrasted them between places with and without otters. Same exact approach. And we also were able to look at these systems before versus after, and after versus before, as otter populations were recovering through time and declining through time. And we've done it in 3 places again. Pretty much the same stuff, just looking at different things. So the first thing, one of the first things we did, this wasn't actually the first, but it was sort of the coolest in a way, work that I did with David Duggins and Cy Simonstead way back in the, back in the 80s. And this was actually David's idea and Cy's idea that there ought to be a difference in production between systems with and without otters, because there is a difference in the abundance of plants or autotrophs in the system. So what we did was we took baby, recently settled mussels and barnacles from Friday Harbor Lab, same species that occurs in the Aleutian Islands. We'd never be able to get a permit today, but then we didn't have to. And we took them out there and we outplanted these things to islands with and without otters, and then we went back and collected them a year later and looked at their growth rates, and we looked at their carbon isotopic signatures to see how much of the carbon that went into the growth of these animals came from phytoplankton versus kelp. That's relatively easy to do out there. And I'm just going to show you one piece of data. These are for the mussels, and what you see here is that the growth rates of mussels in otter-dominated systems, kelp forest type systems, are about twice as high as they are when the otters are absent. And that's almost certainly just because there's a lot more production and a lot more particulate organic, and maybe dissolved organic carbon in the coastal water columns in these otter-dominated systems, and it seems to be fueling a greater secondary production in the filter feeders. These are some data on fish abundance, and I won't go into all the details. I just summarized them all in a single slide here to show you how the catch effort, which is how we measured greenling abundance, and it's virtually impossible to count them or measure them in other ways. So this is a system that we used. And it's about an order of magnitude greater in kelp forests in otter-dominated systems than in systems where otters are absent. These are data on glaucous winged gulls, another consumer in the coastal ecosystem. They feed in the intertidal and the shallow subtidal. And what I've done is I've just separated the data on all the foraging data we have from looking at pellets and from watching them feed into 2 groups, fish and invertebrates. If you go to places where otters are abundant, the gulls are piscivores. They feed mostly on fish, hardly at all on invertebrates. If you go to places where otters are absent, they're virtually exclusively invertebrate feeders. Very few fish, lots of invertebrates. Makes sense, that's what's there, all right? That's probably the explanation. Here are the eagle data. Now these data actually were gathered by Bob Anthony, one of my good friends and colleagues, and who just passed away a little over a year ago. Eagle guy. He was out there working on eagles, and he'd been measuring, looking at the diet of eagles in the Aleutian Islands before, when otters were abundant out there. This was during the 80s, late 80s and early 1990s. And what he found was that there was roughly an even mix of seabirds, fish, and mammals in the diet. A little bit more birds, but more or less even mix. Then the Aleutian Islands otter population collapsed during the 1990s. When it collapsed, the kelp forest system shifted to an urchin-dominated system, went back, we resurveyed all the same nests. There's about 300 and some nests over quite a few islands. And the diet had changed really radically. The fish had become less abundant, mammals had become less abundant, seabirds had become much more abundant in their diet. Bob also had gathered data on contaminants, on persistent organic compounds, in particular DDTs, PCBs, and things like that. And the contaminant levels had gone way up in the eagles as a consequence of this. And that's because now they're eating seabirds. Seabirds are flying all over the North Pacific. They're getting polluted. They're coming back to these places. So that seems to be what's going on. So this interaction actually has an effect on the degree to which these birds are being subjected to potentially damaging anthropogenic contaminants. Okay, the last thing I want to talk about in this context, and almost all of what we've looked at is just a teeny little piece of what we could look at. But we became interested a few years ago in the question of whether this trophic cascade had any significant effect on carbon sequestration from the atmosphere. Because we knew kelp was abundant. We knew kelp, when otters were abundant, kelp was way more abundant than when they weren't. And we knew that kelp were very productive. And we know that they're fueled by carbon dioxide like every other plant. And so we thought, I wonder if there's anything going on there. So this is sort of the logic, okay? And this was actually an idea. This wasn't my idea. We had a new faculty member that moved to Santa Cruz about 6, 7, 8 years ago, Chris Wilmers, and he wanted to talk about ideas of things we might do together. And we were having lunch one day, and he said, have you ever thought about this? And I said, you got to be nuts. I mean, this cannot be going on. I mean, this is not, no way. You know, I mean, the atmosphere and all the gigatons of carbon that are floating around out there just didn't seem like it would. But he'd been thinking about it because he was interested in climate change, and he said, well, I'm not so sure, and so we talked a little bit more, and well, the question was, can sea otters reduce atmospheric carbon dioxide through this trophic cascade? So we talked about it, and I said, it's a lot of work for something that's probably going to turn out to be nothing. But we decided, we can just basically sit down and figure this out from the stuff we've already got. Because all we need to know is how much kelp there is between areas with and without otters. We need to know how much habitat there is. We knew that. We need to know the proportion of the area that's rocky reef kelp habitat. We could figure that out. There's data out there all over the place now where you can map these things. We need to know what the carbon content of the kelp is. That was easy. Matt did that. Other people have done it. Matt was involved with this work. We need to know what primary production of kelp was. People have done a ton of that. We knew that. We didn't have to do any of that. And we needed to know how much carbon dioxide there was in the overlying atmosphere. Well, everybody had done all that. We didn't have to do anything other than just put all the stuff together and just ask the question, does it matter? So we did. And what we found was that it does matter. It matters a lot on a local scale. It doesn't matter on a global scale. It's a little teeny part of the overall world. But on the scale of a local kelp forest and that overlying piece of atmosphere, which admittedly is a conceptual construction. It's not a real thing, but that overlying piece of atmosphere, what we found was that the kelp carbon pool made up a significant proportion of the overlying carbon dioxide in the atmosphere. About 10% of the total overlying carbon dioxide, or about 40% of the increase in carbon dioxide since the beginning of the Industrial Revolution, would be accounted for by having a system that had otters in it compared with ones that didn't. And if you translate that into production, which is higher than standing biomass, so the production of kelp is about 3 to 4 times the standing biomass on a per year basis, this then starts to become really interesting. It starts to become interesting depending upon where that production goes when it goes out of the kelp forest, and we don't know that yet. We know some of it probably goes into the deep sea, maybe a fair amount. Some of it may actually be remineralized fairly quickly. We don't know what happens to it, so that becomes a really important question. But nonetheless, this kelp forest is processing a high proportion of the inorganic carbon in the overlying atmosphere and the bicarbonate CO2 that's in the water in the kelp forest. Depending upon whether otters are there or not, it's very different. The carbon cycle works in a different way. And so this just gives you a sense of what this means. We calculated how much this carbon was worth in the North Pacific from the Aleutian Islands down to Vancouver Island. And we used the European Carbon Exchange, carbon credits on the European Carbon Exchange for making that calculation. So the standing biomass is worth about anywhere from $200 million to about $500 million. And this sort of blew me away because everybody's arguing about shell fisheries and what they're worth, and they're worth nothing compared to this. The sequestration thing is potentially hugely valuable depending upon where this carbon, the production, is going. If it's sequestering into the deep sea and holding there for 100 years before it comes back up to the surface, it could be worth over $1 billion a year. So I think this is a big deal, and it really got me excited at a time in my life when I wasn't sure I'd be excited about too many things anymore. All right, I want to move on and talk about this issue of strong interactions playing out over time and the degree to which those can have evolutionary consequences. What I call eco-evo reciprocity. I don't call it that. Other people have called it that. That's become sort of an important kind of a catchphrase in ecology and evolution in the last 5 or 6 years. People are really interested in the interplay between ecology and evolution. And one of the things that we have come to realize is, and everyone in here who studies evolution knows this now, that evolution can happen really fast. It's not something that takes thousands of years to happen. It can happen in a generation. Okay? I mean, it's been shown in fish selection experiments that in one generation you can actually get selective effects. So evolution can happen very quickly. So we had to figure out what is it that we wanted to look at that had to do with evolution. And it didn't actually really happen this way, but I'm going to tell the story this way because it's easier to understand. We could have looked anywhere in this food web for things where we would expect selective influences, very different kinds or forces of species interactions, depending upon whether otters were present or absent. Where we looked was at the interactions between herbivores and plants. And we did that for a bunch of reasons. One is they're abundant. You can work with them. You don't need permits to do anything with urchins or kelp. You can go out and kill them and sample them and measure things and all this sort of stuff. They don't run around. We already knew a fair amount from terrestrial work and some of the marine work about how marine plants deal with herbivory. And one of the things that we knew, mostly from Mark Hayes, really important sort of pioneering work in this and a few others with him, is that algae don't have a lot of different ways of defending themselves against herbivores. They don't have calcified tissues. They're long-lived. They can't do it in life history. They can't get up and run away like any plant. They can't get up and run away. I mean, they can disperse their spores, and maybe hopefully they'll settle someplace that's good. But basically, the adult plants, once you become a sporophyte, once you become a sporophyte, you're there. So we were pretty sure that if there was a significant modulation of the interaction between herbivores and plants, it probably involved secondary compounds. So then we needed some way to contrast, to perturb the system in a way that would be interesting from an evolutionary perspective. So we simply decided to look at the North Pacific and compare that with the Northwest Pacific, the Northeast Pacific with the Southwest Pacific, because this is where the otters occur. This is the system in which we imagine this trophic cascade having been a strong evolutionary force over quite a bit of time. Otters have been there for 2 to 3 million years in this system, probably a few things before them. We concluded that there wasn't anything of comparable importance in the southern hemisphere. All right, that became really important. In other words, we went down there imagining that this was essentially a two trophic level system with herbivores and plants, but without a predator that was anywhere near as effective as the otters. Now, we knew that wasn't quite true. We knew there were predators there. We knew from work that had been done that these predators could have limiting effects on herbivores. But we also knew from all the published work that those looked like kind of wimpy interactions compared with what was going on here. And I still stand by that. But it's a little bit flaky because it's just a hard thing to really demonstrate. So one of the things we did to begin with was we went to a bunch of places and we looked at the in situ rates of herbivory. And we did that by taking a piece of kelp, putting the kelp on the seafloor, and putting it next to another piece of kelp that was in a cage. So the caged piece of kelp can't get grazed, so that's a control. And the one that's laying next to it out on the seafloor can get grazed. And so the difference between the control, the cage control, and the non-cage control is a measure of in situ herbivory. So this is the difference between the cage control and the non-cage control at Amchitka Island, where otters are abundant. This is the difference over on the far right-hand side at Shemi Island, where otters are absent, and it's radically different, as you can see. I mean, they're just chomping the kelp to pieces here, hardly eating any of it at all here. And these are the rates at 4 sites in marine reserves in New Zealand. And this was very exciting. It may not look exciting to you, but it was super exciting to us because it was significantly greater than this and way significantly less than this. So the next thing we did was we looked at the secondary chemistry, the potential defensive compounds. This is a little bit tricky because what do you look for? Well, we knew pretty much what to look for, and I won't go into all the reasons why. We knew to look for fluorotannins. Fluorotannins are ubiquitous in the marine brown algae, and we already knew that they were defensive, and we had pretty good reason to believe in the North Pacific that they were important. So we measured fluorotannins, and this is the frequency distribution of percent dry weight of fluorotannins across all the common kelps in the North Pacific and all the common kelps and kelp analogs and rockweeds in Australia and New Zealand. And what you see is that it's about an order of magnitude greater in Australia and New Zealand, the overall mean concentration. There are a few that don't have much. There are some that have a lot, but on average it's about 10% dry mass of the plants are fluorotannins. That's a lot of their dry mass. In the North Pacific, there's almost none. These are the common things: Macrocystis, Nereocystis, Laminaria, Pterogophora, the really common things. Agarum, cabrosum is out here. It's a little bit better defended plant. But for the most part, it's really different, the profiles. So when Peter Steinberg, who I worked with on this, he was a student at Santa Cruz, and we got onto this stuff together. And he went to Australia and New Zealand to do a Fulbright before we knew any of this stuff. And the first night he was there, I got a fax back from him, and he said, "You will not believe this. I just walked out on the beach and I can smell the fluorotannins. It's different." And sure enough, it was when we did the analysis. So then the question became, have the herbivores that evolved in the southern hemisphere responded to these defenses, these purported defenses, in any sort of a different way than the ones that evolved in the northern hemisphere? Because they're eating a plant food resource that's very different. So how do we test that? Well, there are all kinds of problems to testing that. And one of them is you can't just put plants out there because plants different in their morphology, and the concentrations of the compounds different among different tissues, and on and on. There are all kinds of problems with doing that. So what we did was we simply extracted the compounds, purified them. It's very easy to do. Even I could be taught to do that. You know, it's really easy to extract and purify fluorotannins. And then we put them in these neutral grazing models. And the neutral grazing model is just ulva, freeze-dried, mixed up in agar, and you take a little disc of of agar with ulva in it, and you put it in with any marine herbivore, snails, urchins, whatever, and they scarf it. So it's a great neutral grazing model, and then we laced it with fluorotannins. We put fluorotannins in different concentrations from different parts of the world and looked at how the herbivores responded. And here's a table from our paper that was published in the mid-90s on this issue. Let me just explain to you. These are California herbivores on the left side of the vertical red line, New Zealand herbivores on the right side of the vertical red line, fluorotannins from southern hemisphere algae above the horizontal red line, fluorotannins from North American algae below the horizontal red line. And the NS's indicate no significant effect of fluorotannins. And the stars indicate a significant deterrence effect. Either a really super strong one when you've got a couple stars, or a significant one, less strong with a single star. And basically, if you kind of squint and look at that, what you see is there's very little deterrence in the southern hemisphere and lots of deterrence in the northern hemisphere. In other words, northern hemisphere herbivores are really sensitive to fluorotannins. Southern hemisphere herbivores are much less sensitive. And it doesn't matter where the stuff comes from. It doesn't matter whether it comes from the northern hemisphere or the southern hemisphere. So that was sort of the last piece, and we boldly proposed this mechanism, this sort of coevolutionary mechanism that explained what we thought was the difference in the way that the plants and herbivores co-evolved in this system, depending upon whether you were in Australia, New Zealand, or in the northern hemisphere. And the basic idea was simply this. In this two trophic level system, Australia, New Zealand, you've got chronic, strong herbivory going on all the time. That has selected for defenses in the plants. They become fluorotannin rich. Those defenses, in turn, have selected for resistance by the herbivores. And so you've had this evolutionary arms race that's been going on down there because of this interaction. And that explains why the plants are so nasty and why the herbivores don't pay a lot of attention, as much attention to that nastiness. At a third trophic level, and it just breaks out all apart. There's no longer any reason for the plants to defend themselves, thus there's no longer any reason for the herbivores to resist those defenses, and that's exactly what we see. Is this true? I don't know. I don't know. All I know is that the patterns are really intriguing because they fit this model. That's all I can tell you. All right? So, now I just want to spend a little bit of time talking about this reciprocity of ecology and evolution, because if these systems in fact have co-evolved in really different ways, that is the plants and the herbivores, then you would expect that to play out in the ecology of the system. So here's one thing that just is really interesting. This just was published last year, maybe 2 years ago now. So these are all the data I showed you before that show the covariation in abundance of herbivores, urchins in this case, and plants, and you see this very hyperbolic type relationship. That is, most of the state space is unoccupied, which is to say that you almost never see plants and herbivores living together in abundance. When herbivores are abundant, plants are rare. When plants are abundant, herbivores are rare. That's the way it is in the northern hemisphere, most places. That's not the way it is in the southern hemisphere. So these are data that were gathered in exactly the same way in the southern hemisphere, and the plants and herbivores live together down there in abundance, often. So I've thought about some other things. This is one of them, something I can't help wonder about. This is stellar sea cow, this wonderful big hydrodamylene sirenian that occurred in the North Pacific and is now gone. So we've come to believe that this interaction between otters, urchins, and kelp been pretty important to the evolution and extinction of the sea cow. I don't have a lot of time to go into this. This is a paper I'm working on right now with Dan Doak. We're doing some modeling work looking at the question of whether the surviving sea cows would have been able to survive if otters were lost from the system, and the answer is almost certainly not. There just wouldn't have been enough there for them to eat. I can go into that in more detail. This is another dimension that we've looked at. I got interested in body size evolution in abalones, mostly through interactions that I had with Dave Lindbergh at UC Berkeley, who's a malacologist and evolutionary biologist, and we got to talking about body size at some point and kind of marveling about how big the abalones were in the North Pacific. And David had been doing some work on abalones, and he pointed out a few things to me. One is that all the tropical abalones are little. All the big abalones in the world are high-latitude species. They live in kelp forest systems. Apparently that's because of the production of food, although it may be for some other reason. And so what we did was we looked at maximum reported body size in all of the paleo records, all the fossil records, and in all extant systems in the world today. And these are just the extant data. So this shows, these are just box plot diagrams of maximum reported body size in abalones for various faunas around the world, the Australian fauna, New Zealand, Indo-Pacific, East African, and so on and so forth. And what you see is that the two very biggest are the ones in the Northeast Pacific. Japan and the Northeast Pacific are the biggest of all abalones. And we imagine that that very well could be because they evolved in an environment where the food was really good. There just wasn't a lot of junk in the food that they had to defend themselves against or learn how to digest or any of those sorts of things. If, in fact, you take kelp from the North Pacific and put some fluorotannins in it and raise red abalones on that fluorotannin-laced kelp, they grow much more slowly. Okay, so the last part, and you're probably wondering how anything could ever be any more speculative than what I've already said, but this is, I'm going to talk more about some of the inter-ecosystem connectivity work that we've done. And that was prompted by these data. Our interest in inter-ecosystem connectivity was prompted by these data. And these are data that we had gathered from the Aleutian Islands on the abundance of otters. We began to recognize in the early 1990s that numbers of animals in the Aleutian Islands were declining. And I didn't believe it until about the mid-90s because there was just no reason to believe that they should be declining. But by the mid-1990s, these open circles are the data that we have from Adak Island where we've been surveying. Pretty clear that the numbers had gone down, so then we went back and resurveyed some other islands and saw virtually the same pattern in all of these other islands. The otters were crashing. And so couldn't help but ask why, because it was completely unexpected. And the conclusion that we came to, and this is a long story, I actually gave a seminar down here about 20 years, 15 years ago on this very topic, but we came to the conclusion that the most likely explanation for these declines was predation by killer whales. And very briefly, this was the evidence. All of a sudden, we started seeing them, killer whales, that is, every day. Hadn't seen them hardly at all for 20 years. All of a sudden, in the early 90s, they showed up. They started eating otters. We saw them eating otters. Had never seen a killer whale eat an otter before 1991, and then we started seeing them. Not a lot, but we did see them, all right? And the rate of attack from our field data, I went back and took all my old field notes and computed rates of attack, and they were highly significantly different, much higher. We had a couple little places that killer whales couldn't get into, but where sea otters lived, and they didn't decline there. So the numbers stayed high in those places. And then we did a bunch of modeling work to ask the question, could it be that what we've seen really is explainable by killer whale predation? And the answer was, without question, it was consistent. So for example, we asked the question, how many mortality events would we expect to see, given the size of our field crew and the number of hours we spent in the field, how many of those events would we expect to see in that time-space sample space, all right, the area and the time that we sampled or were out there working, if killer whales killed every otter in the system that was, in other words, that was responsible for that decline. So what we did was we just parameterized a simple population model, calculated how many deaths it would take on top of those that were occurring beforehand to drive the population down at the observed rate. And then we computed how many deaths would have occurred over the period of this decline, and then we computed how many we would expect to see. And the number that we saw the number that we calculated was that should have occurred was about 50,000. The number we saw was 6. We saw 6. That's pretty bad. I mean, that sort of had me worried, because that's a big difference between 50,000 and 6. But I also knew that we weren't seeing them all, and so when we calculated how many we would expect to see, it turned out to be 5. That had a huge effect on my thinking. Not that I think it's statistically hugely robust, but it showed me that not seeing very many was in fact what we would expect to see. If we'd seen hundreds, there would have been something wrong. But the fact that we only saw 6 meant that that's about what we would expect to see if killer whales were driving this whole thing. So this is a characterization of the way that I see the system having changed as a result of this. Started out as a 3 trophic level system, otters limiting urchins, thus enhancing kelp, and it went to a 4 trophic level system. With the arrival of these killer whales, otters became rare, urchins became abundant, kelps became rare. These are the data on all of these various things, and in fact, it's just exactly what we saw happen. Pretty sure this is right. Just seen too much for it to be anything else. But, of course, that led to the question of why did that happen? Why did these things come in? If in fact we were correct, why is it that the killer whales came in and started eating the otters? And I didn't have any idea why that should be. And I don't have time to go through all of the logic of what led us to this hypothesis. But this led to a paper that was published by Alan Springer at the University of Alaska called the sequential megafaunal collapse hypothesis. And Alan's idea, and I signed on, it's not all Alan's, a bunch of us on this paper, which some of you who are in the marine mammal world will know is extremely controversial and remains so, was that it had to do with whaling. And the idea was that these big killer whales were either predators or scavengers on this huge biomass of great whales that were in the North Pacific. And what happened to that biomass is that it basically disappeared. And it started disappearing right after World War II. And that happened because after Japan was beaten down during the war and Russia was essentially, their economy was destroyed, the United States, General MacArthur, who was in charge of the Pacific theater, told Russia and Japan and helped them basically get their economies back on their feet by fishing and whaling at very high rates, and they did that. And here are the whaling data. So this is from 46 to 53, 8 years later, and then 8 years later up to 1979, the end of commercial whaling. These red dots are the IWC records of whale kills, where and how many occurred. About 500,000 whales were exploited from the North Pacific during that period of time. And by the end of it all, they basically were gone from the North Pacific. Most of the whaling in the end was sperm whales and Breedis whales further south. All the big, the humpbacks and fin whales and sperm whales in the north, in the Aleutian Islands and Bering Sea and North Pacific were pretty much so rare they weren't even worth hunting anymore. So here are the data now that we put together, and these show the number of whale landings within 200 nautical miles of the Aleutian Islands. Starts out 1950, almost none, goes to a peak, and then collapses by 1970, it was over. Here are the otter data. They went crashing down in the 1990s. Here are some data for harbor seals, stellar sea lions, which we also knew had declined. And here are data for harbor seals, which we also knew had declined. So we looked at all this stuff and said, my God, look at this stuff. It looks like these killer whales, just by sort of projecting backward, once the big whales were out of the system, may have just essentially hunted down this food web by going from one species to the next to the next. I don't know if that's true, and we were sensitive to the uncertainties of this. We have done some modeling to ask the question, would the system be sustainable without the big whales? And the answer is no. Basically, there are too many killer whales out there to make a living on marine mammals if the big whales were taken out. So that's one thing we do know, but there's a lot that we don't. So this is the way I see the food web now, and I use this analogy to Darwin's simile of nature as a tangled bank. It's very complicated, this food web. There are lots of important linkages that occur in space and time. As I see it, this is just my perspective on the system, this otter, urchin, kelp cascade is a really important player right in the middle of it all. We have these spin-off effects in the coastal ecosystem that I told you some about. Some of them are bottom-up effects, which are the gray arrows. Some of them are top-down effects, which are the black arrows. Some of them are things that we're pretty sure are going on, which are the solid arrows. Some of them are things that we are less certain are going on, which are the dotted lines, all right? But now I see this system as not being an isolated kelp forest, but one that's really linked by events that are going on in the open sea and very likely through the interactions of great whales and killer whales, and that this system is all tied together in space and time. That is to say that this kelp forest ecosystem was apparently very seriously perturbed by something that didn't even happen in the kelp forest system, and in fact started a half a century earlier, and has now led to changes in things like coastal fish and urchins and kelp, and changes in atmospheric carbon dioxide that had their origin very potentially, not for sure, but potentially 50 years ago with whaling and the interaction between great whales and killer whales. So, just in retrospection, I want to say a couple of things about the major lessons that I see from this 50 years of working in this system. I've told you about the sea otters and kelp and the relationship among these through this trophic cascade. We've looked at the generality and found that it is, in fact, a fairly generally occurring interaction in the areas that we've worked in. We've looked at the knock-on effects through the food web and the coastal ecosystem and found that there are some very strong things going on there. We've tried to look at the evolutionary and adaptive significances of changes in these species interactions and have seen some evidence that there are important interactions going on in that context. We have seen that this system is clearly linked. We may not have all the details right, but whatever happened to the otters, it didn't start in the coastal system. I'm sure of that. It started in the open sea. And so I just want to end about 30 seconds by telling you a little bit about the future, because I'm still trying to do some field work. And so this is work that I'm doing with these three people: Bob Stenick, some of you will know, University of Maine; Doug Rasher, who's a postdoc with Bob and me; and Christy Croker, who's one of our new hires at UCSC, just starting this first year. And we are looking at this thing, which is a crustose coralline alga. That's Clathromorphum nereostratum. And this is a giant. a big one that's probably somewhere between 750 and 1,000 years old. Very long-lived. And these are really cool because they lay down this calcareous record that gives you growth and geochemistry and all kinds of cool stuff that you can look at through time. And so this is a time surrogate for us. Basically, like tree ring people have looked back, using tree rings to look back in time, we're using growth rings in this species to look back in time. I just want to show you a couple really interesting things. So this is a section through a colony from Attu Island, a 136-year record. These is a cross-section. These are the conceptacles. They lay down a layer of conceptacles every year. You can count them just like tree rings. And if you look at the rate of growth of this particular colony through time, it's been virtually constant. I know that Attu Island has been an urchin-dominated state for at least 130 years. I've seen it for 50 years, and I know what the otters were doing for 100 years before that. So I'm pretty sure this was an otter-free system throughout. This is a comparable colony from Ogliuga Island, which is in the Delaross, which is about 400 miles east of Attu. And it's not quite as old. This is a 52-year-old specimen. And what do we see? We see a really high rate of growth, a low rate of growth, and then a high rate of growth recently. What's going on here? Well, I'm pretty sure that this was before the otters colonized Ogliuga. This is during the period that they had colonized and it had become a kelp forest, and then this reflects the period of time after they've collapsed and it's become an urchin barren again. So these are just one cut that we did out on the ship this summer. We're analyzing all this stuff now, but it looks like it's going to be very promising as a surrogate for looking back in time. So I've already run over, as I almost always do. I'll stop with that, and if you'd like to hang out and talk or have questions, happy to answer them. Thank you very much.
Has anybody validated those growth zones?
Yeah.
I looked at a lot of growth zones, I'm not seeing exactly where the ground lines are.
Yeah, it's
And
kind of
I'm
hard
used
to
to
see.
eating with it.
It's kind of hard to see on that. So we have validated them, both Bob counts them, but we've also used dating techniques. We used a uranium thorium dating technique to confirm that we know that they lay down conceptacles every year. They lay down these conceptacles every year. So just like a tree ring, except during bizarre years, it might be super wet or super dry or something, you might get a strange one, but we're pretty sure that that they are laid down every year.
Are they pretty deep or shallow?
These specimens come from about 30 feet.
So they'd be a perfect candidate for pondering or
They will see it. You can see it in there, I'm sure, no question. Yeah, I'm sure you can see it. You know, everything that has carbon in it, that's long-lived, you can see it in other things, I'm sure. We haven't looked, this is not what we're interested in. We may actually use that to confirm the ages on some of them because we know when that started. So we may do some of that. Anything else? Yes.
So in Hawaii, I'm a whale tour guide, and I tell people that the whales are coming back, because that's what I've seen from Noah.
They are.
So with that, would you expect that it should turn back into close to the water?
Maybe. They haven't completely recovered. Certainly in the North Pacific, there aren't anything close to being recovered to what they were. They have significantly recovered. There's no question about that. They have quite a bit. Terminal certainly have increased in combat. Fins not so much, blues not at all, but they have. But on top of that is the problem that it may also be a system that's characterized by hysteresis. That is, it could be that there have been enough behavioral changes in these things that it won't just linearly track backwards, just like the Air Force system did. And so that's a kind of a cop-out. But it's one reason why it's maybe not a very good way to test the hypothesis.
Yeah.
Okay.
Just following on that, is there any hope for recovery for otters in the, you see any signs, any positive signs, any illusions that they're coming back or that they might?
I think it's hopeless. I've seen no signs of their recovering. They are extinct on some of the islands now, some of the smaller islands. I can't imagine what it would take to turn them around. But having said that, throughout my whole life, I've always been surprised, and about half the time I've been wrong, or I thought something was going on, and so, who knows? You know, but just based on what we know about process, or think about process, and what we're seeing in the way of pattern, which is trends, this decline started in 1990, roughly, more or less, in a few years. We're now 2015, and for the last 10 years, they've basically been bottoming out, and we haven't seen any sign of recovery. I had expected the numbers were so depleted that we would start to see the effects, just because I thought they would, be a reproductive problem. We're not seeing any evidence of that either. The reproductive rates have been virtually unchanged throughout this whole period.
So Jim, I had two questions. The first one, right after that, is how do they compare to the post-human exploitation of otter snares? You have those little remnants. So the population level and the evolutions at the end of hunting, are those numbers higher than what you're seeing?
I think the numbers that are out there now are probably higher than they were at that time. But we don't know for sure how many there were. They were thought to be extinct for about 20 or 30 years. There was a period from the early 1900s until about the late 1920s when sea otters were thought to be gone entirely. And then they were rediscovered at Chick Island. That was the first place that they were rediscovered. Then in 1938, they were rediscovered along the Big Sur coast. They were found down there, such a remote place. If somebody knew they were there, but they weren't telling. So I don't think that it's as bad as it was then. So I think they could recover, but it was clear what the cause of the decline was then. And it was easy to manage, and that was people. How do you manage this thing? Maybe with whales, but certainly not going to go out and blow away with killer whales. That's just not going to happen for a whole bunch of obvious reasons. So, to answer your question, I don't see any hope for them recovering, but then,
My second question is, why was it so much more black and white in Southeast Alaska than was in the Aleutians on those perch plots?
because in Southeast Alaska, you get very episodic recruitment of the urchins. And so these urchin populations that are unexploited by otters are virtually all big animals. When the otters come through, they just hammer them. In the Aleutians, there's very strong recruitment. So you've got a lot of little ones. And those little ones are not eaten as commonly by the otters. In fact, otters won't eat an urchin that's less than about 25 millimeters of test diameter. So that's why you see those different cellular patterns there. When they come through Southeast Alaska, it's just night and day. Just to give you an example, when I was diving about 20 years ago in Sitka Sound, before the otters came through, there were nothing but red urchins. We did not have one single kelp plant in any of our quadrats or transects that we sampled. We sampled hundreds and hundreds of them. Not one kelp plant. Urchins everywhere. We went back 3, 4, 5 years ago now, I guess it was. Resampled all those sites. Otters that moved through. We did not find one urchin. Not one single urchin plant. All gone. And there was kelp just everywhere. So it's really, really a strong and rapid interaction in that place. But I think the difference has to do with recruitment. Why that is, I don't know. It may be oceanographic. I think it has a little bit to do with oceanography. If you don't have this constant Ekman transport in the Aleutian Islands where stuff's just being swept away from the coastal system. I also think it has to do with the fact that the oceanic islands, and so there are these deep water populations of urchins that constantly are just conveying up from deep water to shallow water where there's more food. And along the continental shelf, you've got this big band of sediment that's a barrier to deep water species individually from the shallow water. So I think those are some of the potential explanations, but those are just sort of the way treatments. Yeah.
So over time, what are the orca population numbers moving?
There are lots of them now. There are about the same densities of orcas in the North Pacific that there are in the North Atlantic, that there are in the whole southern oceans. So on that basis, I'm sort of imagining that they haven't changed a lot, but they very well may have changed a lot. One of the things that has been speculated about in the North Pacific was that the big impact on orcas was not on the provisioning of a resource for scavenging. So when whaling started, two things occurred. One was, well, three things. They killed a whale. The orca
float out there for about a day before the processors came along and dragged them on board. So the catchers would kill them, they gas them, so they float them. When they kill them, they use an exploding harpoon, which advertises the kill. Thing can really hear for a long ways. And so it advertises the kill. Things floating there, it's undefended. And so there's a lot of literature to indicate that the killer whales really honed in on that. So that may have actually had a huge facilitating effect on the killer whale population. And then when industrial whaling was mandated to be illegal by the International Whaling Convention, all of a sudden that disappeared instantly. And so that might be what shocked the system so much. And all of a sudden things didn't have this food resource anymore. So how might that many reads have sort of developed that idea? And it makes sense to me.
But we don't know, since the decline of the great whales, what part of the population is.
We do. You know, we don't. We know about how many transient killer whales are out there based on killer whales, based on visual IDs, and the discovery curve, and they live a long time, and that discovery curve is still growing a little bit.